this video is going to wrap up the last of our biotechnology section that we started in class. So let's step through it. Okay, so we know about restriction enzymes. We know that they're used to cut DNA up into pieces. And we know that we can cut up DNA from pretty much anything and compare it. Well, why would we want to do that? Well, there are lots of applications for the comparison of DNA. Let's start with things like forensics. It's one of the ways that we can compare a suspect or many suspects' DNA to DNA that we found on the actual crime scene, on the victim, on the weapon, etc., etc., etc. It's a way to form a link. We can use it to do medical diagnostics. We can look at the DNA that gives rise to one particular condition versus one that does not give rise to that particular condition and make some comparisons there. Without comparative DNA, Maury Povich and Jerry Springer wouldn't kind of be out of work because kind of known for their paternity testing that they do on their shows. But it's a very viable way to kind of figure out who the father of a child is, dad 1 versus dad 2. We can look at evolutionary relationships. If there are things in common on a genetic level, it normally means that organisms or species are interrelated or linked or maybe evolved one from the other. So how do we compare DNA? We need to separate the pieces out. We know we can use restriction enzymes to cut them up into sections, but now we need to take those sections and separate them based on size. We do it by running them through a gel, and that gel can be made of agarose, which comes from algae, or it can be made out of a substance called polyacrylamide. But it gives you a gel that has microscopic pores in it. So think of Jell-O but harder and not as tasty. And we can use that, apply an electrical current to it, and do what we call a gel electrophoresis. So that's another one of those tools or techniques that we would have as a biotechnologist. And here, here's a picture of what that gel with those pieces would look like. The little dark regions that you're seeing, that would be the actual DNA. All right, so how does this gel electrophoresis work? We know it's a method to separate DNA, but we're going to do it based on two things, charge and size. DNA has an overall negative charge, because remember all of those oxygens on the phosphate groups? That makes the entire thing negative. So that means when it's in an electrical field, it is going to naturally move in the direction of the positive electrode, wherever that positive electrode will be. So we set up our DNA on a gel. We normally make a little depression for the DNA, which is called a well. The DNA is placed into that well, and we pour a buffered liquid solution over the gel, and we put it in a chamber, and we attach a positive electrode, which will be on this end, and a negative electrode, which would be on that end. We flip the switch, and we let it run for about an hour. And this is what you would see. You would see the DNA pieces traveling down the gel. And they separate based on size. The bigger the piece, the closer to the top or the closer to the well it will be found. The smaller the piece, the further away it is going to be from the well. All right? So again, smaller pieces travel further, so they move further down the gel, whereas larger pieces travel slower and they tend to get stuck closer to the top part of the gel. Right, so here's a mock-up just to show you. This would be our power source right here, negative cathode, positive anode. Here we are loading our DNA into the wells. You let them run for a little bit, and then you see this separation. You see these bands. Okay, and then after a while, you know, you'll see really good separation the longer you leave it. But after a while, you know that the shorter molecules will be on the bottom of the gel, whereas the longer molecules will be up at the top of the gel. But DNA is clear, so you have to stain it. And the stain that we use is ethidium bromide, and ethidium bromide binds to DNA, and then it fluoresces under UV light. So we stain it, we let the stain sit and take, and then we put it on a UV light or box light source, and you turn the light on, and then you'll see these stains glow. They'll fluoresce. Okay, so in terms of evolutionary relationships, like I said, we could compare DNA samples from different organisms as a way to measure how closely related they are. And a lot of the cladograms that we studied when we looked at ecology and we looked at evolution is based on information that we got from gels. So based on the separation and how many bands they have in common and things like that. Okay, for medical diagnostics, like I said, you could compare DNA of a normal allele to DNA of a diseased allele and look at how big they are, how small they are, where they're found, how far down the gel it would separate, that kind of thing. Right, so this is just another example underneath that forensics bracket. So in this one, we're looking at the defendant's blood versus blood found on the defendant's clothes, jeans, and shirt versus the victim's blood. And you can see some patterns that they have in common right here. And this is called a DNA fingerprint. It's what you get when you compare DNA banding between different individuals. Okay, and you just get these unique patterns that can give you some information. All right. So on the DNA level, let's talk about why each person's DNA pattern is different. A lot of our genome is the same from individual to individual. But what allows us to have unique patterns in terms of DNA is the fact that we have these sections of junk DNA. Remember that junk DNA doesn't actually code for proteins, and they tend to be made up of repeated patterns like CAT and GCC, and there's some others. And each individual may have different numbers of these repeats. So we'll all have like CGG, for example, but I might have CGG repeated 27 times, whereas somebody else may have CGG repeated 5 times. And that would make a distinction or a difference between my DNA versus their DNA. There are also many sites on our 23 chromosomes with different repeat patterns. So on my chromosome 1, I might have certain repeat patterns that someone else would not have on their chromosome 1. And that helps to identify who I am. It ties my DNA specifically to me because of the uniqueness of it. Okay. So now that leads us to talk about what we call RFLPs, restriction, fragment, length, polymorphism. This is the differences in DNA between individuals, and it's a way of looking at what those differences are. So it's a change in DNA sequence which affects where restriction enzymes are going to cut. And because of these changes, it's going to create fragment sizes that are different from one individual to another, but it's also going to create a pattern that's different from one individual to another. Because if I don't have certain restriction enzyme sites on my DNA, restriction enzymes can't cut them. Or if I have more than somebody else, my pieces will be smaller because those enzymes would have cut more times on me than on someone else. So it just gives us these unique bandings, and you can literally look at one person versus another, or one allele versus another allele. Okay, polymorphisms, again, these are differences between individuals at the DNA level, and many of these differences accumulate in our junk DNA, in our DNA that doesn't actually code for any particular protein. It's just kind of there to make us unique. All right, and then here's another example of you're just looking at a gel. This one actually is kind of cool. This is a gel that was based on the O.J. Simpson trial when they suspected him of murdering his wife Nicole and her friend Ron, I think was his name, Ron Goldman or Ronald Goldman, whatever his name was. So you can see that they're looking at samples from the crime scene. They have 3 different samples from the crime scene. And then we have a standard. The standard just tells us actual sizes. We know for a fact that this one is this big and that one is that big. So we have something to compare these two. And then here is blood from the suspect, in this case O.J. Simpson, and blood from victim number 1, which was his wife or ex-wife maybe, and then blood from victim number 2, which would have been Ron Goldman. And you can just see where the patterns line up. So O.J.'s blood here matches sample 3 from the crime scene and sample 1 from the crime scene, whereas Nicole's blood matches sample 2 from the crime scene. It doesn't seem like Ronald Goldman's blood matches any of these particular samples. Okay, so we know that we can use bacteria to make lots of copies of DNA. But it would be so much easier if we didn't have to wait and depend on bacteria to first pick up the plasmid with our gene that we want made, and then to actually replicate. Yes, it's a fast process, but it would be even better if the process could be even faster. There is a way to make the process faster. We can copy DNA without plasmids through a method or an instrument called PCR. And PCR stands for polymerase chain reaction. It's a method for making many, many copies of a specific segment or section of DNA. And you only need one cell of DNA to start with. So you don't have to worry about the bacteria, you don't have to worry about the plasmids, you don't have to worry about housing them and making sure the temperature is right and not killing the whole colony or anything like that. You just need one sample of DNA and you can start. Okay, so let's talk about what the PCR process is. It's a way of copying DNA pretty much in a test tube, or at least in an endorphin tube. And that's what an endorphin tube looks like. So what you need is a template strand, a strand that you're going to build your messenger RNA, well, your DNA from, sorry. And you need an enzyme that will be able to build DNA. Well, why not use the same enzyme that our bodies already use in the form of DNA polymerase 3? You will also need some nucleotides because polymerase can't just build DNA out of, thin air. You need adenine and thymine and cytosine and guanine. Remember, they're going to come with their own energy source, so they're actually going to come in the form of nucleotides, ATP, GTP, CTP, and TTP. And then you need a little bit of a primer because, again, polymerase can't attach DNA to nothing. They need something to do to attach the DNA that they're going to build. They have to put it on something. Then the last thing you need is the instrument that actually does this, which is called a thermocycler. And they look like this. The newer ones look more modern, more sleek maybe than these do. But pretty much they're a very expensive box that heats up and cools down because that's the essence of what it does. Okay. Now, one of the most important parts of this whole process are the primers. If the primers are not right, you're not going to get a good workable piece of DNA. So in order to have a primer, it means that you need to know a little bit of the sequence. You have to know a little bit of the sequence of the piece of DNA that you're using, if it's AATC or CCTG or whatever that sequence is. Because you're going to order these primers or build these primers based on the sequence of the piece of DNA that you're trying to copy. So you start off with a long piece of DNA and you copy a specified shorter section of the DNA, and then the primers will define the section of DNA that's going to be cloned. So if you look at the picture, if you only want this region of DNA, then you're going to have at least that much primer so that polymerase can build the rest of it in both directions. So how it works is there are 20 to 30 cycles in 3 very specific steps, and it takes about 30 seconds per step. So we'll step through those. All right, so what do you need to do? Well, in your Eppendorf tube, you're going to put your DNA, you're going to put your enzyme, your primer, and your nucleotide. And then you're going to insert it into your thermocycler and set your heat settings. The first thing you're going to need to do is you're going to need to denature the DNA. And that means that you're going to have to heat it up to about 90 degrees Celsius to separate the strands out. Then you're going to have to anneal the DNA, cool it down so that it can hybridize, it can bond with the primers, and DNA can be built. So we're going to hybridize and extend all in one step. But do we have a problem? We may have a problem. 90 degrees Celsius is pretty high, especially for a human enzyme. And enzymes are proteins, so chances are at 90 degrees our DNA polymerase may not really work the way it should. So ponder on that for a minute. So how do we fix it? Because it is a problem. Human DNA polymerase is not going to work. It's going to denature. It's going to unwind because it is a protein. It is going to destroy those bonds and just cause the polymerase to unravel. So it means that every single time we did PCR, for every single step, you'd have to put in a new enzyme because after a while the old one would get worn out. It would be denatured. Well, that's very impractical and really not efficient. So instead of doing that, what you need to do is find an enzyme that can withstand that 90 degrees Celsius. And there is one. It comes in the form of what we call tag polymerase, and it is from bacteria that live in very hot conditions, like hot springs, for example. But the polymerase works the same way that DNA polymerase works. It just adds nucleotides or bases based on whatever our template strand is. So this enzyme is even more efficient than DNA polymerase, the human version, and it can withstand the temperature that we need it to withstand. Okay, so now that we know we can just make DNA as we need to, another good thing to have would be something called a DNA library, and we do have those. DNA libraries are what you get when you cut up all nuclear DNA from many cells of an organism using a restriction enzyme. You clone all of the fragments into plasmids at the same time. It's a technique that we call shotgun cloning. And then you store the collection of DNA fragments. And you can do that in a petri dish or, there are lots of different methods for storing them now. But essentially what you do is you have a whole bunch of different kinds of DNA from lots of different organisms, and you know exactly where to find them. It's like a library. So how do you make a library? Well, the first thing you got to do is figure out which restriction enzymes you're going to use and use that to cut out your gene that you're interested in. Then you're going to engineer the plasmid by placing the gene inside of the plasmid and also attaching what we call a selectable marker in a screenable system so that it'll do something when the bacteria takes it up, like it might fluoresce or it might give off a smell of some kind. And then you insert all of the DNA fragments into several plasmids all at once, and then you let the bacteria clone the plasmids. And there you have it. You have your DNA library. All right. So once the plasmids have been cloned, because every time this cell goes through binary fission, every new daughter cell is going to have that particular gene already in them. So then all you do is you take your bacterial colonies and you plate them, and you store and copy all of the genes that the organisms produce. So you just literally label them. This is human insulin, this is human growth factor, this is human HCG, or whatever it is. So it means that now it's easier for us to find our genes of interest because we have them in these DNA libraries. But how do we find them? Well, to find them, you need to know some of your genes' sequence. So if you know just a section of those nitrogen bases, like how they align in the protein, you can then back translate the protein into DNA. All right, so let's say that again. If you know the sequence of the protein, so, leucine, lysine, cytosine, whatever those amino acids are, then you can guess a part of the DNA sequence because, there are only so many codes that would code for those individual amino acids. If you know the mRNA code, then you can take that and back translate into the DNA code. So we can literally go from protein to DNA. If you have a sequence of a similar gene from another organism, you can use part of that sequence, probably because the genetic code is universal. So it's going to be the same amino acids, which means it's going to be the same nitrogen bases, regardless of whether it's found in an amoeba versus a giraffe. All right, what are the problems? Well, in the human genome library, we're going to have junk DNA as well as coding DNA. And because we have so much junk DNA, our genomic library has more junk than it has genes. So you're going to have to sift through all of the junk in order to find the gene that you are interested in. So how do we fix that? Well, you clean up the junk. If you want to clone a human gene into a bacteria, you can't do that easily because human genes have introns. Bacteria don't have introns. So that would cause a problem, or it would cause an issue for sure. So how do you clean up the junk? It's simple. You don't start with DNA. DNA has junk in it. But remember, through protein synthesis, we can make mRNA, and after we fixed up the mRNA, we don't have junk because one of those 3 processing steps is to cut out the introns and leave just the exons. So if we start with messenger RNA, you can make a copy of the gene that has no introns in it whatsoever. But, I mean, that's great. It's great that we won't have to worry about the introns, but at the end, we're still going to need DNA. We can't put messenger RNA into a plasmid. We need DNA to go into the plasmid. So what do? We have to go backwards. We have to go from RNA to DNA. And there's special enzymes that can do that. They're called reverse transcriptase enzymes. And we got them from viruses that utilize RNA as their genetic material. And after this video, you're watching a video on viruses, which is your last one for the year, and we'll talk a little bit about reverse transcriptase enzymes in there as well. The names that we give to these viruses, we call them retroviruses because they're doing the opposite. They're starting with RNA and using enzymes to make DNA from RNA. But this works perfectly for us in our issue because if we start with RNA, we don't have to worry about junk. But we still need some DNA. So we reverse transcribe the process. It's like we're doing we're doing the transcription part of protein synthesis in the opposite way from the way it normally goes. We're using RNA as our template. We're using reverse transcriptase to build a DNA molecule. Okay, so that allows us to build a special kind of library called a cDNA library or a copy DNA library. And in that library, we're only going to have coding sequences of expressed genes. And we got those sequences because we took them straight from mRNA. Remember, anything that is in mRNA is going to be expressed as a protein. There's no junk in it. There's no extra material. There's no nothing that isn't important to the protein. So once we got it from mRNA, we have exactly just the genes needed for the making of that particular protein. And again, that's done through the enzyme reverse transcriptase that can take RNA to DNA, or start with RNA and make DNA, and we found these in retroviruses. This allows us to clone our new DNA, the one that just has the gene, into our plasmid, and we can then do whatever it is we need to do with it. So what's cool about this is we don't have to worry about making a mistake. There's some extra stuff in this gene that's not going to be turned into a protein. And it reduces on the need to edit the DNA for expression in bacteria. We have exactly what's going to make our gene of choice, insulin, HGC, whatever. All right, I hope that wasn't too difficult to understand. Sometimes this can sound very abstract just because, it's hard to break it down to a practical level, a level that everyone gets. But as usual, if you have questions, we can address those in class. So I'll see you guys on Monday. Bye.